My name is Alice. I am a PhD candidate at the University of Southern California with Suzanne Edmonds. I study largely toxicology and kind of how we use different methods and ideas from evolutionary biology to kind of answer questions related to toxicology. And what I'm doing in this work here is looking at how copper tolerance and heat tolerance may or may not be related in this particular species of copepod, Tigriopus californicus. So the best place to start about this is kind of just to think very broadly about multiple stressors first. And that's just to say, when we have multiple stressors that are impacting an organism, we're going to have multiple possible outcomes. Those are depicted here in this figure, where we have stressors A and B independently shown in gray, and the height of the bar represents kind of the negative impact they have on an organism. The first scenario or outcome we have with multiple stressors is just an additive one, where the effects of A and B together is just the sum of their individual effects. We have antagonistic stressors, and so that's when A and B together are less than what we would think the additive impact is. And then we have synergistic stressors, and so this is when A and B together are worse than the predicted additive effect. Traditionally, kind of synergistic stressors get the most attention in this area because they present the greatest potential for harm to an environment or an ecosystem. But for my purposes, I'm super interested in antagonistic stressors. That's because two stressors kind of not being as bad as we thought is kind of a contradiction, at least biologically, and comes as a surprise. So with antagonistic stressors, what I want to know is what specific mechanisms are going to cause these interactions, and what combinations of stressors are going to give us this antagonistic outcome. There are a couple of different proposed mechanisms that will lead to antagonistic interactions. The one that I'm studying here is cross-tolerance, which is depicted in this figure over here. And so basically what this is when you get a brief exposure to one stressor, that increases that organism's tolerance to a second stressor. So it's just a brief pulse that results in an increase to that second stressor that might be totally different, that only lasts for a short period of time, but there is a definitive boost when it happens. Cross-tolerance is thought to be underlaid by two stressors either activating the same stress response mechanisms, like on a molecular level, or activating the same signaling pathways. And so what I'm doing in my work is evaluating whether or not different stressors can generate or get cross-tolerance in our study species, which is this little orange crustacean shown here called Tigriopus californicus. So this little orange bug is a copepod, and so this is a small crustacean that's found in these splash pools along the intertidal of the Pacific coast of North America, reaching as far south as Baja California and all the way up to Alaska. So this is a field site here, and we found organisms in those gross little pools shown there. The reason why we like this organism is because it is super easy to maintain in the lab, and because it harbors a lot of interesting features genetically. One of those features being that it features very strong signal, I should say, of local adaptation to different conditions among various populations, even those that might be quite close together. The best studied kind of pattern of local adaptation has to do with thermal tolerance, where populations from southern areas like San Diego and Baja California are better at dealing with high heat than those from more northern areas. So this latitudinal decline in thermal tolerance is very well studied and kind of to be expected based on what we know about the temperature decline in the northern hemisphere. And we've also found signs of local adaptation that also track with latitude with other factors like salinity and oxygen, or hypoxia, I should say. But what was super interesting is some previous work in our lab identified a possible latitudinal decline in copper tolerance, which is shown here. So this plot shows you the copper tolerance of different populations plotted along with the average temperature, or maximum temperature, I should say, of each population's collection site. You can kind of already see without knowing which population is which, that we see an association with warmer temperature and better copper tolerance. And then if you go in and check which population is which, we see that there's a cluster here, or there's one population from Central California, so a more northern one, and then this group here, these are all from Southern California. So it seems like there might be some kind of association of copper tolerance with the latitudinal gradient of temperature, which doesn't really make any sense, largely because we don't have any knowledge of copper pollution increasing with decreasing latitude. So there's no reason to think that San Diego waters are necessarily more polluted than those by San Francisco or up in Washington. state. So my hypothesis is that this potential relationship between latitude and temperature and copper tolerance has to do with overlapping stress defense mechanisms. So the way that thermal or heat stress generates kind of problems on a molecular level is by speeding up the rate of metabolism that generates reactive oxygen species that cause oxidative stress. Copper will also initiate oxidative stress by generating ROS, although through a different mechanism. So if we have this potential overlap of the stress defense mechanism, that could explain both this pattern and potentially lead to cross-tolerance. So that leads me to the research questions for my project. Firstly, just wanted to go in and kind of evaluate that little latitudinal gradient a little bit more thoroughly and see if we can identify it in more populations covering a greater span of their range. Second, wanted to find out, is there cross-tolerance between copper and heat? And then kind of as a sub-question within that, if there is cross-tolerance, does the order of sequence of stressors matter? Do we get cross-tolerance going in both directions? So what I did is I went out and sampled from 7 different sites in California, going as far south as La Jolla, which is down in San Diego, and as far north as Bodega Bay, which is about an hour and a half north of San Francisco. So went to all these places, collected animals, and started a culture in the lab. And what I did was set them through a series of stress experiments. The first type being single stressor, where animals were exposed to either a range of copper concentrations or a range of high temperatures to gauge tolerance in that single stressor situation. And then to set up sequential stressor experiments, where animals either got a very small dose of high heat or a very small dose of copper, and then followed by the reciprocal stressors full experiment. So those single stressor ones are just to look at tolerance in isolation, and those sequential ones are designed to look for cross-tolerance explicitly. And what we found is, first of all, that both thermal tolerance and copper tolerance are correlated with latitude. The heat data is shown on the left, and the copper data is shown on the right. And you can see in both cases, elevated tolerance occurs at lower latitudes, so from warmer areas. And so we have that negative correlation between tolerance and latitude for both stressors. The error bars are much larger for that copper data on the right, largely because there's quite a range of concentrations being used. I used 12 different ones, versus just only about 7 or 8 degrees difference in that temperature data. So this is nice because we kind of reconfirm that thermal latitudinal gradient that we already knew about, and we have a little bit more evidence for that copper latitudinal gradient as well. When you plot both copper tolerance and temperature tolerance or thermal tolerance together, we see a nice correlation between those two things, although there is a bit of variation between those populations. So nice confirmation of those initial findings that we had earlier in the lab. Now the most interesting stuff or the most exciting stuff comes when we look at the difference in those tolerance values between our single stressor experiments and our sequential stressors. So in this one, this is looking at copper tolerance with and without doing a heat shock, where the standard value is shown with a circle, and the triangular value represents copper tolerance after receiving a heat shock. And you can see across the board in every population, we do get that boost in copper tolerance, in some cases quite substantially. There doesn't seem to be any sort of pattern, though, in terms of which populations received a greater boost in terms of their relationship to latitude. So this here is quite strongly an example of cross-tolerance, where we did get that boost by doing that brief heat shock prior to testing. The reciprocal scenario, though, of when we do that copper shock before thermal testing gave us a slightly different answer, where copper shock actually ended up reducing thermal tolerance. So here, again, the standard value, the median lethal temperature in the single stressor experiment is shown with the circle, and then the copper shock version of that, where they got a copper dose before heat testing, is shown with the square. And again, we get a drop in thermal tolerance when we have that copper shock in each population. So this is the opposite of cross-tolerance. You could maybe argue that this is leaning more towards an additive interaction or synergistic interaction, but from this alone, it's hard to tell. But what this is saying, at least to me, is that cross-tolerance for this particular set of stressors only goes in one way, where we get heat first and then copper. So that suggests that there's some kind of difference that's a little bit more complex in terms of how the molecular response mechanisms work for both these two stressors. So maybe that heat one is a little bit more broad because heat initiates kind of molecular stress in multiple ways versus just how copper generates reactive oxygen species through one specific reaction called the Fenton reaction. So there's potentially some very interesting molecular questions we can move forwards with based off these results. And so the next phase of the project, which I don't have data for you today, but what I am actively working on, is evaluating gene expression responses in our most divergent populations, so Bodega Bay and La Jolla, and see how they respond to those different stressors to see where there is and is not overlap in terms of the different enzymes and proteins and other molecules that are being used to deal with these sort of difficult molecular situations. So the takeaways here, first of all, we have that positive correlation between copper and heat tolerance, which is really exciting. Second, a stressor sequence really, really matters for cross tolerance, particularly in this case where we have one sort of precise stressor and one more generic. And then lastly, that heat response mechanism might be a bit broader than copper's. So in the interest of time, I'll just wrap it up there. So throw my acknowledgements up in terms of the friends at the lab and my devoted team of undergrads, as well as the funding and people who helped let me go to their different places to collect copepods. And if there are any questions, I'd be happy to take them, and you can access contact and my most recent publication up here. Yeah.
I'm wondering, were you using a set of different temperatures for the populations, or were all of them treated at the same temperature?
So each population was exposed to the same range of temperatures, going from, I believe, 35 up to 42 Celsius. Yeah. Okay. If there's no one else, maybe I'll just move on to adjust for the little timing issue to start. Okay.
Thanks for coming to my talk in this session. My name is Kendra Gardner, and I will be providing insights into lumpfish diet combating salmon lice in aquaculture. So first things first, what is aquaculture? So it's defined as the farming of aquatic organisms, including fish, mollusks, crustaceans, and aquatic plants under controlled conditions. So in 2022, Canadian aquaculture, excluding Newfoundland, produced approximately $1.4 billion, and Newfoundland alone produced around $200 million. So from these numbers, we can tell that this is a large industry with a high economic value. However, there are detrimental issues, and one of the more large ones to the industry is sea lice infestations. So sea lice are marine ectoparasitic copepods that belong in the Caligidae family. They attach the rim of their cephalothorax to the host fish's skin and then use their modified thoracic appendages, here it's labeled free thoracic segment, to stay attached through section. So salmon lice belong to the genus Lepiophthirus and are much more likely to attach to a host fish from the salmonid family, hence the common name salmon louse. And their infestations tend to be more severe in the late summer and early fall, just because their generation time is shorter when the water temperature is warmer, so they reproduce more, so there's just more of them to attack the fish. So when an aquaculture farm experiences an infestation, the lice will feed on the host fish's skin and mucous tissues, resulting in decreased fish welfare. And there are also environmental concerns, as the larvae can travel through the netting of the aquaculture cages into surrounding waters, where they can then infect the wild populations as they swim by. These infestations also have a significant economic impact. Depending on the condition of the infested fish, the company may not be able to sell them, resulting in loss of income. And additionally, there are multiple treatments out there, but some of them can get very costly and be quite expensive. So what are these treatments? One of the more frequently used ones in the past was chemotherapeutic treatments. However, this method can be controversial, and this is the more expensive method. In 2010, the average cost was $630 million globally. And the most common method of this was an in-feed treatment, as I'm demonstrating here. So they use the insecticide slice, put it in the salmon feed, and then it just kind of repels the salmon lice from attaching. However, over time, the sea lice evolved and became genetically resistant to the insecticide, and also the public just wasn't very keen on insecticides being used in their food production. So scientists looked elsewhere, and we ended on biological controls such as cleaner fish. So cleaner fish are a type of fish that clean another fish, often by removing ectoparasites or other parasites. And this has become the preferred method because it's less likely that the sea lice can become resistant to the cleaner fish, and if they do, then there's often coevolution going on, so the fish kind of combat that. So the most commonly used fish in aquaculture is the lumpfish, this cute little blue guy here. Lumpfish are cold water marine fish with a broad natural habitat, so they're native to the North Atlantic Ocean. And they eat prey items that fit in their mouths using a gulping technique, so they're highly opportunistic generalist feeders. So for current research, studies have shown that cleaner fish do lower parasite infection rates and negatively affect their population dynamics. However, majority of research on cleaner fish has been done in tropical species, but there is more research being done on lumpfish as a cleaner fish. So Jessica Roy, who is a previous graduate student in my lab, conducted a general dietary analysis on lumpfish, and my thesis project expands on her project and what she found to further our understanding of lumpfish diet and how it changes. So morphological species analysis is a method very common in dietary analyses. However, it can be very time-consuming and also not really reliable depending on the digestion level of the prey items. It can lead to misidentification or just missing species entirely. So to combat this, DNA metabarcoding using dietary DNA, or ddNA, is becoming a more popular method as it's more reliable and can actually find and identify organisms that would have been entirely missed otherwise just from them being digested. And for those who don't know, DNA barcoding uses standardized short genetic sequences and compares the sequences to a database to make an identification. So all this leads to my research objectives, which were to assess relationships between diet composition and lumpfish size within Atlantic salmon cages in Newfoundland, and then also to improve the BOLD database for marine invertebrate species in Atlantic Canada, specifically around aquaculture cages and farms. So I predicted smaller lumpfish would consume more of the salmon lice and biofouling organisms, which are just organisms attached to the netting and the floating pipes of the cages. And then larger lumpfish will consume more of the feed pellets and Norwegian crow. So how did I do all this? All fieldwork was conducted in the Olive Cove farm site near Hermitage Bay in southern Newfoundland, which Cook Aquaculture Inc. has permits for. And their scuba divers collected a total of 69 lumpfish from 9 cages at that farm site, so in Olive Cove. For both stomach removal in the field and stomach dissections in the lab, we followed strict protocols to maintain ddNA quality and also reduce contamination. So to remove any DNA and DNAs that we didn't want around, we used a 10% bleach bath in the field and 10% hydrochloric acid solution in the lab. And then we followed that with DNA grade water, and this was applied to all surfaces, equipment, tools, everything that was being used. I euthanized the lumpfish to cut out their stomachs and remove them, and then they were preserved in 95% ethanol and stored in a negative 20 degrees Celsius freezer just to maintain the integrity. For stomach dissections, we carefully emptied out the stomach contents, and then once that was done, we began sorting the prey items into predetermined categories. To maintain identification normality, we had different scales. So one was for identification confidence, the other was for digestion level, and that was just to maintain normality among the technicians. For the homogenization of the DNA, I used liquid nitrogen and a mortar and pestle to grind the stomach contents into a fine powder, and then I put that powder into reinforced centrifuge tubes, so we didn't lose any samples, thankfully, and then put those tubes in a fast prep bead beater just to ensure they were fully homogenized. And DNA was extracted using the Quiamp Fast DNA stool mini kit. For all metabarcoding work, we're using a primer pair that targets the BR5 fragment on the mitochondrial cytochrome oxidase 1 gene. The forward primer targets freshwater benthic invertebrates, and the reverse primer targets tropical arthropods, and Jessica Roy had used these primers, so we know that they target we know they amplify the target ddNA. And then all sequencing was done at the AAC Genomics facility at University of Guelph. To amplify the extracted DNA, I did PCR, so polymerase chain reactions, on all my samples, and then to ensure that the PCR worked properly, I ran agarose gel electrophoresis assays of the amplified ddNA, and there's a nice photo of one of them that worked well. I also collected 84 voucher specimens from a total of 7 different cages across 2 farms. These specimens came from the netting of the cages and then also underneath the floating pipes, and I just grabbed them with my hand, so collected by hand. I morphologically identified each specimen using multiple identification reference guides, and also I used the World Register of Marine Species, or WORMS. I did also receive some training by Ed Hendricks at the Canadian Museum of Nature on certain families of amphipods, which was extremely helpful. There are so many amphipods around. The middle one is just all amphipods, so is the bottom right, so they were very abundant. And then out of the 84, 31 voucher specimens were submitted for sequencing and photographing at the Center for Biodiversity and Genomics Center at the University of Guelph. And then once I have their sequences, I'm going to send them to BOLD to update their database. So I just have some preliminary results to show. So this graph here is showing the percent frequency of occurrence, or percent foo, as I like to say because it's fun, of the voucher specimens. Essentially, it's just showing how often a genus appeared as a percentage of the total number of voucher specimens identified, which would be 84 for this case. The most common genera of the voucher specimens were Caprella, Mytilus, and Obelia, and they're all different families as well. For the percent foo of prey items, again, it's just showing how often a prey item appeared in a stomach as a percentage of the total 69 stomachs examined. The most common prey categories were other, salmon pellet, and Caprellidae. And Caprellidae is a family of amphipods. The other category just refers to organisms that didn't quite fit into any of the other categories, or if they were too digested to make an identification. And then this slide here is just comparing the percent of prey categories that I collected against what Jessica Roy collected back in 2018. And I just wanted to show this because we both collected lumpfish from the same aquaculture farm. And there is some differences in the diet composition, which I find to be interesting. She found more general amphipods, krill, and benthic invertebrates than I did, but I had way more other or unidentifiable organisms, feed pellets as a whole, and then also ctenophore and jellyfish and sea lice. So one of my little theories for the variation is that I had a lot more pellets. So I think the lumpfish were just so full from these nice, formulated feed that they just didn't eat anything else. And that may not have been the case when she had collected them, since she had less of the feed. Also, abiotic factors could play a role, such as water temperature. When I collected my samples in 2023, the water was abnormally warm for that time of year, and I had multiple people telling me that, just because of different heat waves throughout the summer and early fall. So I'm going to be looking into how that may play a factor in the diet composition. I conducted principal coordinate analysis, or PCOA, on all my stomach data, using this code here for those interested. And then I created graphs to visualize the differences in the lumpfish diet based on their total length and their weight as 2 separate parameters. So this graph here is showing the color clusters based on the total length, and there aren't any significant clusters based on the color. That would kind of indicate that total length may not play a large role or impact the diet composition, and this was statistically supported by my Permanova results, which were insignificant in the sense of stats analysis. And then I had very similar results to the weight, so again, there's not really any clear significant clusters based on the weight categories, and again, it was supported by the Permanova, unfortunately for me. But so for metabarcoding, out of my 69 samples, 65 were submitted for sequencing, and I have 54 results so far. I'll be running my sequencing results through MetaWorks, which is a pipeline that will be able to provide taxonomic assignment. However, I haven't been able to run it through the pipeline yet just because of file formatting issues. The file I have, the header is different, so we're working on that now. But there's just a little snippet of what my sequencing files look like. So in conclusion, while there is variation in diet among lumpfish, there are no significant differences between lumpfish length or weight and diet composition based on the morphological results alone. I'm hoping that will change with the metabarcoding results. My next steps include analyzing the metabarcoding once we fix the file format issue, and then performing other multivariate analyses to look at other variables that may affect diet composition. So I'd just like to acknowledge NSERC and MyTax for their funding, Cook Aquaculture for allowing me to come to their aquaculture farms and take all their lumpfish away from them, and also the Canadian Museum of Nature with a special shout out to Ed for taking the time to help train me, and then just members and colleagues of my lab, and then my family and friends. And thank you for listening. Okay. Were there any questions at all? No? Yeah.
Forgive me if you said it in your talk, but how popular is the lumpfish method for cleaner fish in general?
It's gaining popularity. They're used in pretty much all Norwegian and Scandinavian farms. So then we've started using them in Canadian aquaculture. I know the aquaculture company I worked with, they use lumpfish. I assume the other companies would also be using the fish just because it is a cold water fish. There are other cleaner fish that could be used, but they're not as ideal for Canadian conditions just based on temperature. Yeah. Okay, I think for time I'll wrap it up, but feel free to come find me if you have any other questions. Thank you.
Hi, everyone. I think everything's working. Yeah, thank you. My name is Annette Kang, and I'm a PhD candidate in Corey Moreau's lab at Cornell University, and I'm super excited to share with you my current stage of progress on ecological niche modeling of Paraponera clavata. So many of us are interested in understanding an organism's ecological niche and what variables contribute to its occupied role in space. And so we broadly start with an organism's fundamental niche, which is the entire set of conditions under which an organism can survive and reproduce. And so it's fairly representative of an organism under ideal conditions. And then we'll zoom into the realized niche space to identify the set of conditions that are actually utilized in actual space and role occupied in the real world. And so we'll think about those conditions as abiotic and biotic factors. And so abiotic factors will include things such as soil type, soil composition, precipitation, temperature, and pH of the environment. And then when we go into the biotic sector, we think of interactions with other organisms that can manifest in the form of parasitism, predation, and competition. And so ecological niche modeling, which I'll be abbreviating in E&M in some parts of the slides, it utilizes the abiotic factors and conditions as input information to predict the distribution of species across geographic space and time. And so the abiotic inputs, and ultimately it allows us to broadly estimate the abiotic tolerance, limits, and range of the species in question or that we're curious about, and also estimate the changes in habitat suitability. So whether it remains within the known habitats or whether it forecasts some wildly different distribution into currently unknown habitats that we are just not aware of. And so more importantly, E&Ms really allow us to visualize these niche shifts through predictive mapping and allow us to consider how changing/different conditions can really affect niche parameters of the organisms. So why is this important? Well, it's critical to see how our incredibly biodiverse world reflects the rapidly changing climate and landscapes of our planet. And so as we qualitatively and quantitatively record data on rapid habitat loss and fragmentation, as well as extreme climate and weather events, E&Ms under these predictive projections are capable of realistically reflecting the negative ecological consequences, and it can really serve as an important tool in conservation measures for endangered flora and fauna. Now, with that said, conservation focus has a tendency to fall on charismatic megafauna, while smaller and lesser known organisms tend to get overlooked, even if they are incredibly ecologically important. Which brings me to ants. And so, given their incredibly diverse portfolio of morphologies, behaviors, evolutionary histories, and life histories, much of ecological niche modeling work within the ant world itself, too, has really focused on invasive dynamics and their ecological impacts. And so E&M work on ant species without these attachments to invasive dynamics hasn't seen as much attention as a result. And so I don't want to say that that's a bad thing, because from these past studies, we know that there are some general abiotic factors and general niche requirements that are really shaping the ecological niche of ants. So things such as precipitation, temperature, and soil types. And so it really gives us a fair foundation to really kind of think about how these variables will continue to or change in terms of contribution to predictive mapping as we kind of see this very climate unstable world. And so keeping that in mind, I hope to investigate the contribution of these more commonly seen variables, as well as hopefully seeing other abiotic factors towards defining the ecological niche of the giant bullet ant, Paraponera clavata. And so given its widespread distribution across the hyperdiverse Central and South American tropics, its generalist predator feeding preferences, and its close dependency to the trees of tropical forests for foraging and nesting, amongst other things, I believe that Paraponera clavata makes for an excellent model for E&M work. In addition to what we know about Paraponera's ecological niche, it's important to note that it's the only extant species in the subfamily Paraponerinae. Modeling and visualizing any changes in this monotypic species distributions gives us an estimated foresight and can better prepare us for any conservation measures that may need to be taken. And so, while a previous study from Murphy and Breed in 2007 has shown the possible distribution shifts modeled under specific environmental variables, we've seen a lot of recent advancement in modeling technology and coding and stuff. And so, access to many more occurrence records on top of this really allows us to revisit predictive distribution modeling for P. clavata with a more robust sampling. And so environmental data and monitoring have also seen huge improvements as spatial analysis instruments and tools have become more advanced. And so the resolution of climate and landscape change data, for example, have become increasingly sharp. And I think this reflects the attention and urgency of needing high resolution data in the climate unstable world that we live in and that these bullet ants live in. And so it really gives us the opportunity to take advantage of these improvements. And now I'll dive into some of my guiding questions as I've kind of started thinking about ecological niche modeling in bullet ants. So for one, which abiotic variables contribute most to Paraponera clavata's ecological niche? Will we see the three same general variable contributions, or might we see something else pop up that will be really pleasantly surprising and telling? And then second, how will Paraponera clavata's known distribution shift under changing environmental projections? So what are some unknown habitats that it might start expanding into? We never know. If it comes to North America, it's a little terrifying. Y'all have never been stung by one, have you? Okay, so the process of ecological niche modeling, this is kind of the broad framework, but I'll go into it one by one. And so starting with the occurrence records, I used GBIF to find this data. And so we have occurrence records of totaling to 2,989 that have been taken from multiple natural history collections from across the US and across South America. And on top of that, I also took research grade iNaturalist observations into these records. At this point though, E&Ms, as we move forward, E&Ms really require handling of large spatial and environmental data across varying formats and information. So each step of the workflow kind of includes like a bias correction method or like any sort of cropping and shaping, essentially. And so at this stage, accounting for the spatial biases that can occur from these occurrence records, I spatially thinned out these 2,989 records to account for proximity to biological research stations, highways, and generally areas with easy access for human traffic. And so that leaves me with 124 occurrence records. And then now going into the environmental variable layers. So a total of 24 environmental variables were chosen with matching year ranges and high resolutions. 21 of bioclimatic variables were taken from the CHELSA data set, while 3 landscape variables were taken from the Global Land Analysis and Discovery Lab from the University of Maryland. And finally, going into the third step, which is data management and input correction. So this is probably the most correction-heavy stage of this framework. As I mentioned, there's 24 environmental variables, and we have to make sure we're standardizing each one at every layer. And so this is just an example of me cropping each layer so that it's within my defined study region. This took me like 2 weeks, so I know that might be a lot for some people, but I'm very proud of how pretty it looks. Yeah. But looking at that, I now have to think about how these 24 environmental variables could possibly be covariing because I'm working with such a multivariate data set. And so identifying covariance with this multivariate data set started with an initial PCA visualization. And so running these 24 variables in a PCA, you can see from this scree plot that at least at 60% of the resulting explained variance across all the dimensions are captured in the first 2 dimensions. And so using and utilizing specifically those 2 dimensions, we then plot the variables across these two dimensions as a way to visualize any covariance among the variables. And so I know there's a lot going on, but I'll zoom in next. So color clustering, for one, denotes variable contribution scores across the 2 dimensions. But looking closely at the arrows and paths of some of these variables, we can actually see that some are directly overlapping one another as they increase together. And so that gives us kind of a visual start to kind of contemplating what variables we want to carry on as we go through this framework. So I'm going to show you 3 different pairs of covariant, or what's looked to be visually covariant variables. So we have the mean monthly precipitation amount of the wettest quarter, as well as the mean monthly precipitation amount of the warmest quarter. And so between this pair with the diurnal air temperature range and mean daily maximum air temperature of the warmest month, and the last pair, which is looking at air temperature related variables, I'm going to have to make some decisions and choose one that I've go through the process of figuring out which of these variables are ones that should be carried on to the next one, and then there'll be another stage of checking for covariance with the variance inflation factor later on when I actually have a trained model. Now, next steps. I am still in this stage, but next steps include what I'm currently doing, which is a matrix exponential spatial specification, or more easily said as MESS. So I'm running a MESS analysis right now. And this is the stage where you actually have occurrence records and environmental layers coming together to see if, again, there's any biases, to make sure that once you're in the process of training your model, you want to account for a flexible spatial decay. But ultimately, it just allows you to hopefully get a more improved model and model fit and prediction down the line. And the step after that will be to train my model, tuning and selection, and the ultimate goal of that is to identify the variable contributions to the actual model. And then following that are the projected distributions. And so projected distributions will be under climate projections of the CMIP5 and CMIP6 model. And so these are varying different models across 2 different year chunks. So 2041 to 2070 will be one of the projected distributions, and then something further out from that would be 2071 to 2100. And so that's also data that I can get from the CHALSA database. I think thinking about implications of working on an ecological niche modeling product for Paraponera clavata is following model conclusions. I think there's an importance in kind of breaking down how this pipeline can be applied to other ants in terms of E&M work for them. And so whether that be in invasive dynamics or just generalized like other ants that have niche similarities with bullet ants, or if they're generalist predators, there's a lot of applications there to think about how we can really perfect or really manage this pipeline of E&Ms to fit ants as a model. I think something else that's dawned on me a lot is how important natural history collections and specimen preservations are in generating this presence data. And so when I say presence data, it's because where they are occurring. And so one of the biggest pitfall of E&Ms is that there's a lack of absence data. So we know where things are, but we don't know where things aren't. And so perhaps using these model conclusions, it can start a conversation about how we can start addressing absence data collection in ants specifically. And it can manifest then into a tool in ant biodiversity monitoring. Yeah. So that's the end of my talk, but I'd like to acknowledge my advisor, Corey Moreau, and all the Moreau Lab minions that are here. Thank you. And of course, Mark Buckner, who was my modeling guru. Like, he really helped walk me through all of this, and that's really incredibly helpful, and as well as Justin Schneider. Yeah, thank you so much.
I didn't catch that by quote. So you mean you identify those covariants, those pairs, bio3 and bio6. Do you mean those pairs are correlated and they impose combined importance into your modeling?
Yeah, so because they have, I'll show you maybe this one. So it's really hard to see it, I'm sorry, but they're right overlapping each other. And so at this point, when you're training the model, what it will sense is like double the signal from that direction, essentially. And so, yes, they are essentially giving the same, they are positively covarying, and so they are increasing together. And so we want to account for that. And so it's not bad that they're going in a specific direction. It's bad that they're going in a specific direction together. And so now we have to parse out which variable amongst those three pairs should be taken into account and continued on into the model training aspect.
So all your variables are in this pod.
Yes.
But don't you worry about this collinearity?
Yeah, so I'll be taking, I also did a correlation matrix to kind of think about correlation aspects of this, but we will account for that post-model training, but also the variance inflation factor. That's going to really test if any of these covarying relationships affect the model itself. Yeah. Any other questions that I might be able to answer? No? Okay, awesome. I'm gonna